Um, yes, so thank you to the organizers for this opportunity. I'm a final year PhD candidate in the lab of Corinne Maurice at McGill, and today I'll be presenting some of the work I've been doing over the past year or so, where I'm modeling gut virome diversity, dynamics, and function across 12 infant cohorts. And I'll just begin by acknowledging my lab and funding sources, and with that, we can jump right in. So as you may already know, the human gut microbiota consists of trillions of bacteria, viruses, fungi, and archaea, each of which play important roles in maintaining human health, whether it's helping us metabolize drugs, train our immune system, or digest foods. And phages, or bacteriophages, really dominate the viral component of the gut microbiota called the gut virome. And in adults, studies have shown how the gut virome really is unique to each individual, forming a fingerprint, and is present at a ratio of, and these phages are present at a ratio of about 1 to 1 with their bacterial hosts, reaching abundances of around 10 to the 10 per gram of feces. So really quite abundant in the gut. But if we look at early life and when the gut microbiota really begins to assemble, studies have shown that the gut microbiota assembles according to a defined developmental program where for the gut bacterium, facultative anaerobes really dominate first at birth, and these are slowly replaced by obligate anaerobes as the infant ages and they change their diet. But of course, we're all here for the phages today, so let's get into the virome side of things. And studies have shown that virus-like particles, or VLPs, are detectable somewhere after the first few weeks of life. So that's what I'm showing in this figure here. All these green dots are VLPs, most of which are phages. And you can see how at birth, in this cohort of infants, there are really no VLPs. At 1 month, suddenly, millions of VLPs. And studies have hypothesized that it's prophage induction of these pioneering gut bacteria that's really responsible for populating the gut virome after birth. And similar to the bacterium, there are also shifts in phage kind of abundances and replication cycles with age, and I'll get into that shortly. But all of these studies who have really characterized the gut virome in infants have all done so with their own cohorts and all with their own approaches and pipelines and things like that. So what I'll be presenting today is a more cohesive or comprehensive analysis of 12 previously published gut virome studies using a kind of the same approach for all of them to get a comprehensive overview of gut virome development and phage function during early life. And as I mentioned earlier, the analyses I'll be presenting today is based on 12 cohorts, 6 longitudinal and 6 cross-sectional cohorts that you could see on the map here. And these are really the only cohorts I could find who studied the free gut virome of infants in early life. And it's made up of 999 healthy infants, couldn't find the last infant to make it 1,000, and about 1,800 samples overall. And the infants are sampled regularly. So here I'm just showing you a breakdown of when the samples are obtained. But infants are sampled from birth through 38 months of age, with most of the sampling being done in the first year of life, but some studies continuing to track the infants towards the end. And I won't get into all the details, but I'm happy to answer questions later. We did run all these raw sequencing reads, through our standard existing kind of computational pipeline. We did some protein prediction and annotation as well with different databases, read mapping, host prediction, and a bunch of ecological analyses in R. So that's what I'm going to be going over today. But if you have questions about any of these steps, happy to answer those. So the first thing we wanted to do, we had these 1,800 samples, is to describe virome diversity in early life and get a more comprehensive overview of what that could look like. And whenever we were kind of looking at diversity or some of the other metrics that I'll be presenting today, we used GAMs, which are generalized additive mixed models, as our approach to model a response variable, in this case diversity, across data sets while controlling for specific effects. So GAMs allow us to control for things like sex, birth mode, feeding status, the age of the infant, the cohort, things like that. So removing all these potentially confounding variables, what are we seeing in terms of trend? And when we look at alpha diversity, which is a measure of within sample diversity, we see that for richness, which is just a measure of the number of viral OTU, so the number of viral sequences present in a sample, we see an increase in richness significantly during the first 7 months or so, and then it stabilizes till the end of this study period. And you'll notice on the y-axis here, it says partial effect, and some of these numbers are even negative. So this is how a lot of the plots that I'll present today will look. This is what we get after running a GAM, where it's not signifying directly that there were negative 0.4 viral OTUs at birth. Of course, that doesn't make sense. You can't have negative viruses in your gut. But rather what this kind of signifies is a relative number after controlling and for confounders such as sex, birth mode, cohort, things like that. This is the relative trend in richness over time. And we saw a significant increase in the first 7 months, and then it stabilized. And if we look at another diversity metric called Shannon's index, this is a measure of richness, but also evenness. We see that's pretty much a flat line over the whole study period. So what this would suggest is that although richness is increasing during the first 7 months or so, there's a higher evenness early on, which is then lost when there's this increasing richness. And that's kind of confirmed by the pretty flat Shannon diversity. And we could also zoom into the model a bit further and say, okay, well, after we kind of control for, after we adjust for sex or delivery mode and studies and things like that, what is the model adjusted richness according to delivery mode or sex? And we could see that there was a significantly higher model adjusted viral richness in females than males overall. What this means, I'm not quite sure, but it's just something interesting to note. So this is a measure of, as I mentioned, within sample diversity, alpha diversity, but another way we could look at our samples is by comparing them to each other. And whenever we compare samples between each other, it's called, we're measuring beta diversity. And typically when we look at beta diversity or distances between samples and we generate plots, we get a lot of plots that look like this, and you may already be familiar with them, but I'll just quickly run through this type of principal components or analysis plot, where each dot represents a different sample from a different infant on a different day. And the closer two samples are to each other, the closer two dots are to each other, this would indicate that these two samples are more related to each other than those that are really far apart. So when we plot all our samples in this 2D space, and we could color them by age, we could start to see that there's some clustering or some trend in how the samples maybe move in this 2D space by age, but also by cohort as well. So we wanted to quantify that a bit further. And we could run what's called a primernova, it's just a statistical test to see that sure enough, there is a significant kind of clustering of these samples, not only by study, but also by age. And this was really interesting because we saw that age does play a role now in gut virome development, both within a sample and across samples, but we could start to ask questions of how things are changing with age. So that's what we set out to do next, and you might recall the title of my talk was From Chaos to Convergence, so let's talk about the chaos first. So from chaos, we wanted to see how fast the infant gut virome is changing during this 2 or 3 year sampling period. And to do that, we really subsetted our 12 studies, just focusing on 6, specifically the 6 who had longitudinal data, meaning we tracked the same infants over time. And we defined what's called the virome developmental velocity, or VDV, for infant I at time point t as the distance between the sample from the infant today versus their previous time point divided by the time. And this velocity or speed, a simpler analogy might be if I'm measuring the time it takes me to drive from Montreal to Toronto, I might take the distance that I would drive around 600 kilometers, maybe from Montreal to Toronto, and divide it by the time it takes me to get there, maybe 5 hours on a good day. And that would be kind of my average speed, so my average velocity to get there. And when we look at this virome developmental velocity in the infants over time, and we run a GAM once again to control for some of the confounding effects, we see that there's really the highest virome developmental velocity early on in life, and this is significantly associated with age, where it's quite high in the first 6 or 8 months and then really stabilizes and stays kind of lower. So this is the really chaotic kind of portion of virome development, the first 8 months. And you might recall from one of my earlier slides that we also showed how the first 7 months or so is also the same period where we see a rapid increase in virome richness. So this rapid increase in richness also leads to really significant and important changes between samples during the same period as well. But all of this chaos in the first 8 months or so, how do things stabilize and do they stabilize? So let's get to the convergence side of things. And to look at convergence of samples, we could use a metric called beta dispersion. And this is a measure of variability in community composition among samples within a group. So if we were to group all the 1-month-old infants together, the 2-month-old, 3-month-old, and so on, we could look at how far each of these samples are from kind of the average sample at that time point. And if they're very far, it would be, very dispersed data, very high beta dispersion. And as things kind of stabilize and converge, you would expect that the beta dispersion would decrease. And this is exactly what we saw, where if we look at the green line, so excuse me, looking at how the virome composition looks when we look at the hosts of these phages, so who these phages infect, we see a significant and negative association of beta dispersion with age. But at the VOTU level, so just the raw sequence level, the viral OTU level, there's really no significant association between beta dispersion and age. And what this would suggest is that despite the rapid and kind of chaotic development of the virome in early life, virome composition among infants, when you consider the host that these phages infect, really converges to a common state by around 3 years of age. So from chaos to convergence, that's how we get there. But now that we start to see there's this perhaps predictable nature of virome development, we wanted to see if we could predict an infant's age from the composition of their virome. And yes, this is somewhat of a technical feat just to see, like, can we do it? And sure enough, you'll see that we can. But also an age predictor like this could be used to then kind of identify the most important groups of phages for healthy virome development and see how structured this development is. So to do that, we split our 1,800 samples into a 70-30% trained test split, and then we trained a random forest regression model with these data. And this is what it looked like here, where if we look at the predicted age of the infant on the y-axis versus the actual age of the infant on the x-axis, and in the perfect scenario, the model would return a one-to-one, a perfect 45-degree line, which is what that black line kind of traces. On our training data, you see the model performs quite well. And then once we introduce our test data set, the model still performs quite well and is able to somewhat accurately predict infant age until maybe around 20 months or so. And then it kind of loses its accuracy. And whenever we train models like this, whether they're random forests or other regression models or classifiers, we typically report metrics such as R² and root mean squared error. And one thing that you'll maybe notice right away is that the RMSE, so the root mean squared error on the test data set, is incredibly high. It's around 5 months, right? And that's kind of alarming because it looks like the model is really doing quite well early on, but then it loses its accuracy, right? So we were interested in seeing if there's another way we could maybe more or better classify the error rate of the model to account for this change in error over time. And to do that, we just took a sliding window approach where we had this kind of 3-month sliding window that we would slowly move and look at the average error rate within each window. And that's what the blue line is tracing here. It's the average root mean squared error within each 3-month window. And you'll see that the model error stays quite low during the first 20, 21 months or so, and then it really begins to skyrocket, which is what we saw in the other plot as well. And this corresponds with the period of highest sampling as well. We had the most samples, especially in the first 12 or so months. That's what these bars are showing. And as we have less samples, of course, the model struggles to get any meaningful trend from the data. But also, as the communities kind of converge and stabilize, the model isn't able to necessarily differentiate a 20-month-old virome from a 26-month-old virome, just because things kind of stabilize and there's not much more development happening. And we could zoom into the model a bit further as well and see which groups of phages or the host that they target are appearing and disappearing and when. So that's what I'm showing here is a group of we grouped phages by their hosts and called them PHFs, these phage host families. And we could see these different PHFs that appear and disappear at different ages. So for example, phages targeting Bifidobacteriaceae are very present, very highly abundant in the first 7 or 9 months of life. And these are bacteria, they're bacterial hosts that are very important in feeding milk and formula. So it would make sense that the phages that target these bacteria would be there at the same time as their bacterial host. Whereas phages that target strict anaerobes like Bacteroideaceae or Rickenolaceae come in a bit later, as their hosts also come in a bit later once an infant is weaned. So this was really exciting to see that the trends in gut virome development that we're seeing here mirror what's been shown in other studies with how the gut bacteriome develops. Switching gears a little bit now, I spoke a bit about phage life cycle at the start, So in the gut, most phages have either a lytic or temperate lifestyle, and I don't need to tell this audience what this means, but I'll leave this figure up here as a quick refresher on the two main different life cycles of phages in the gut. And studies have hypothesized that temperate phages are really the most abundant and active in the infant gut microbiome, excuse me, and that they're induced during the birthing process with some kind of unknown signal. So we could predict viral OTU lifestyle using computational tools. And when we plot the abundance or the cumulative relative abundance of temperate phages over time and model that, we see a significant and important decrease in temperate phage abundance over time, indicating that there's this negative association with age, reaching levels that are quite low and similar to what's seen in adults as well. So this was exciting that we could confirm that as well. And one last thing I'll quickly go over in my final few minutes. We looked a bit at functional capacity of these phages. So phages, of course, can carry auxiliary metabolic genes, which are genes that might impact host metabolism but have no direct effect on phage replication. And studies haven't really looked at AMGs in the developing infant gut virome, so we wanted to do that. And after running our viral OTUs and all the 800,000 or so ORFs through our annotation pipeline, we could look at the metabolism categories according to the KEGG metabolism classification system. And we start to see some interesting trends where if we look at the abundance of these different metabolism categories over time in the gut virome, you'll see that AMGs who are important or relevant for glycan metabolism or amino acid metabolism are most abundant in the first year of life, where some other AMGs like lipid metabolism or energy metabolism come in a bit later. So still working through this data. We did some enrichment analyses, but I don't have time to present unless there are questions on that. And with that, I'll just wrap up. So today I showed you how we characterized gut virome development in 999 infants, healthy infants during the first 3 years of life, how the gut virome assembles rapidly and chaotically at first and then becomes more stable and similar with time, and how infant age can be accurately predicted from the gut virome composition alone, most accurately during the first 20 months. And I will just briefly mention that I'm wrapping up my PhD in the next few months, so I'm open to work and looking for next opportunities. And if you have any thing that might be interesting, maybe we could connect on LinkedIn or by email. And I'm happy to take any questions. Thanks very much.